I'm going to talk about the elevated immune response in octopus references under ocean acidification and warming conditions. Introduction. Since the start of the Industrial Revolution, human activities such as burning fossil fuels and coal have increased the concentration of greenhouse gas in the atmosphere, and how increasing carbon dioxide concentration affects the ocean. The ocean acts as a sink for carbon dioxide in the atmosphere, and it also absorbs heat from the planet. And the ocean has absorbed about 1/3 of the anthropogenic carbon dioxide since the Industrial Revolution, and it has resulted a 26% increase in ocean acidity, and with a 250% increase in acidity prediction by the end of the century. And it also rise the ocean surface temperature by 0.85 degrees Celsius. And if we don't make concerted efforts to cut the greenhouse gas emission, it is expected to increase 1 to 3 degrees by the end of the century. And what is the purpose of this study? And the study is trying to understand the immune response of develops to climate change conditions. And our expectation is an increase in overall immune response due to the stressful environment conditions. And what is the importance of octopus in the food chain? Well, they can be found in many depths and marine habitats, and and the octopus are not only prey but also predators for many other species, and they are an important prey for seals, sharks, whales, and seabirds, and they are an indispensable link between trophic levels in many ecosystems. Material and method. In this study, they use octopus luminescence to do the experiment. And what do we know about octopus luminescence? And they are a commonly occurring shallow-water octopus in the North America West Coast, and they can be found from central Alaska to northern Mexico. And they eat small crabs, bivalves, mollusks, and fishes. And their adult weight is about 100 grams to 160 grams. And where the octopus were collected in this study? The octopus was collected in the Driftwood Park near the Canada-United States border. And how the octopus was collected? The octopus was collected by scuba, and after they were collected, they were transported to the lab. In the lab, they record the sex and the weight of each octopus, and each was put into the individual tank before they are ready to do the experiment. And only octopus with a mass of 150 grams were used because they didn't want to sacrifice any octopus after drawing enough blood for study. And by chance, there were no female study were used because none of them met the mass requirement. And to study how climate change, like ocean acidification or ocean warming, will impact the octopus, there are 4 immune system parameters in octopus references to examine. And the 4 immune system parameters are: 1, the hemocyte count. Hemocyte is a blood cell that plays a role in the immune system of invertebrates. And in human body, white blood cells are similar to the hemocyte in the invertebrate. and there is a normal range of concentration for white blood cells, and so is hemocytes. Either an increase or decrease of the normal range may suggest a stress response or illness. 2. The hemocyte phagocytosis activity. Phagocytosis is a critical activity which can protect itself from infectious and non-infectious environmental particles. And 3. The plasma lysozyme activity. Lysozyme is an enzyme that can damage or kill bacteria by breaking down their cell wall. And 4. The spark cell production. It is generally generated by immune system to kill invading microorganisms. And here is the holding tank design. The tank system can maintain the proper temperature and the partial pressure of carbon dioxide and partial pressure of oxygen. And there are 4 treatment groups with different scenarios. And each octopus was randomly assigned to one of these 4 treatment tanks. And blood collection. After 20 to 20 days of the experiment, each was anesthetized until the octopus was unresponsive. Anesthetized means a process that makes you least sensitive to pain. And the target volume of blood was 0.5 milliliters. And after the blood collection, the blood was used to perform the 4 immune system parameters analysis. The result. And after a 3 weeks exposure to elevated partial pressure of carbon dioxide and or temperature, there was a significant difference on the hemocyte concentration and total phagocytosis per volume of blood. You can see that in the same temperature, in different partial pressure of carbon dioxide, there are big difference in the total hemocyte concentration and the total phagocytosis. And there was no effects of carbon dioxide, temperature, or the combination of effects on the proportion of phagocytosis cells, production of spark cell and onion per volume blood, or lysozyme activity. And the purpose of this study is to understand the immune response of developed to climate change conditions. And now we know that exposed to elevated partial pressure of carbon dioxide will increase the blood hemocyte count and the phagocytosis activity. And discussion. The stress response is widely conserved between Texas and may significantly contribute to adaptation. And more study on octopus phagocytosis physiology, especially longer term and throughout the different life stages, are necessary to better understand how this important ecological group will respond to future ocean conditions. Okay, this is the end of my talk.
Okay, I think your PPT looked very different compared to last time, and you added images, icons, and many bullets to make me read the slides easily, and you explained some difficult terms. But some result and discussion slide is a little bit still working.
You use many pictures and slide easy to read, but the slide, some slide too much page